Hi guys, this is Ivor Cummins here, aka Fat Emperor, author of Eat Rich, Live Long, and I'm going to do an Adapt Live chat for you guys, and it'll be on the topic of cholesterol. So hope you find it interesting. So I'm just showing here on the screen a root cause diagram for coronary vascular disease, a disease that everyone's concerned about, of course, and it shows many causes. It is multi-factor. Sure, insulin resistance syndrome or metabolic syndrome is a major causal system that drives oxidative stress and endothelial damage in your arteries, leads to heart disease. But there are many other causes as well, and I won't go through them all here. I've gone through them in other talks. But today we'll talk a little around LDL and LDL particle counts, ApoB or LDLP, goes by a few different names. And if you have high LDL particle counts, that may very well be a problem and will accelerate atherosclerosis. But there will also be scenarios where someone is very healthy with higher counts and may not have an issue at all. So the jury is somewhat out. So I'll talk a little about it today. Now, I would be of the opinion from research that if you don't have any other drivers or root causes of coronary vascular disease in your system, and if you're very healthy, you might be able to have a higher particle count with no deleterious implications. But if you add a high LDL particle count to any or all, and I have not done the full list here of root causes of endothelial distress and inflammation, if you add a higher particle count with some of these problems, you may get an accelerating effect towards cardiovascular disease or atherosclerosis. So this is one way of viewing it. Are you completely healthy with a physiologic higher level of particles not really being a problem? Or are you accelerating coronary vascular disease by having a high particle count driven up by insulin resistance syndrome or interacting synergistically with problems attacking your arteries, and therefore you've got this accelerating effect? So this is an interesting question. So I would tend to focus on the root causes rather than the physiologic particles. But again, that will be a personal decision. And you can certainly bring down your particle count on a low carb diet by just tweaking your dietary inputs as we've laid out in our book. Mainly replacing saturated fat with monounsaturated and bringing up the healthy carbs a little higher percentage in your diet. It's not really rocket science for most. So now we'll go into this LDL particle count challenge and put it into a little bit of context. You know, it's not really an independent driver, even though it's very much involved in the atherosclerotic process. It is very dependent on other aspects of your physiology and your health. So I look first at calcification of Western men, and this is the MESA study. And just to note that very high calcification in the simple CAC scan indicates you can have up to 20 times the risk of future heart attacks or heart events as a person who is a low score or a zero score. So it's one of the most powerful tests in the world to tell you your current level of inflammatory atherosclerotic disease, and hence it predicts future events with an incredible degree of precision compared to other tests or blood markers. So here we see on the left the Western people, and as they go from their 40s up to their 80s, around half of the population in the green oval here, up to 50 percentile, get very little calcification. And generally they have very few events, age standardized, because of course they are not driving the atherosclerotic disease process so hard and they don't calcify. But you'll see in the red oblong up here, 5 to 10% of the top calcifiers are riddled with disease. And this is where most of the events occur. Not exclusively, but the top 10% get 40% of the heart attacks, and so on. So it would be really interesting to look at another population that is not so affected by calcification or heart disease. And the Samain men we'll take as an example because we have some data on those guys. We're going to look first at what kinds of things have they got in their favor, biometrics and other states of health. So I pulled out here a few things. They have very low insulin levels, very low blood glucose, no diabetic physiology whatsoever, it appears, no hypertension, and their blood pressure doesn't increase with age. They've no central obesity, unsurprisingly, and they've great omega-3 to omega-6 ratio in their diet, and there are many more advantages they have from their environment, but I've just pulled out some top Pareto items. Many of you will realize that this list means they have no metabolic syndrome or hyperinsulinemia syndrome, which is kind of a very problematic type of state to be in for heart disease. It's not the only thing. So these guys are really good. And boom, that's the calcification taken from around 700 people of all ages. So we've got the full age compare here with the Western men. And as you can see, they have bugger all calcification. And they have some of the lowest rates of heart disease in the world, as you would expect. So people could say these are genetic unicorns. These are weird people, you know, that came from some bizarre genetic lineage. But the fact is that the Kitavans have similarly very low rates of heart disease, and they have all of the same environmental advantages we show on our list, and some. The other thing to note about these people, and the reason I bring them up, both the diverse Kitavans and the Semaine, very different genetic people, they happen to have ApoB levels measured quite extensively. And their ApoB levels, or LDL particle counts, are around the same average as the US people over here, or actually slightly higher, it would appear from the data. So it shows that ApoB, or particle count, you've got to be very careful looking at any one marker. You know, it's a multifactorial disease. So we'll go on. I'm going to tell you that this list of very advantageous things for your health, which will really help with heart disease if you focus on them, I'm putting a target sign there so you can see this list coming up in future slides in the next 20 minutes. And be mindful of this list. It's not exclusive list of things you need to do. There are more, but it's a pretty important list. So I gave the Cholesterol Conundrum lecture back in 2014. It was quite popular, and I described all of how the lipoprotein system worked and residence time of LDLs and how it was there to deliver triglyceride energy to your muscle and your fat, and basically all around that in detail. So you can Google it if you want to see the first cholesterol presentation I gave. But this one will go on to more detailed questions. I'll just illustrate why we're interested in this question, because many studies indicate that, as you can see here, people with bad total to HDL ratios, or triglyceride to HDL is similar, when they're high or in the middle range, it indicates they're insulin resistant or they may have some other issues that could affect their vasculature and its permeability and its tendency towards atherosclerosis. And as we can see for these groups of people, certainly a higher ApoB or LDLP, the two are kind of quasi-equivalent, a higher level means a significantly higher risk. So we've got to acknowledge ApoB, LDLP as a significant and quite potent risk factor. However, we've also got to be very cognizant that in the same study, the people with the low ratios indicating insulin sensitivity and indicating better vascular health generally, for those guys, the higher ApoB certainly didn't cause more hazard for cardiovascular events. It actually trended towards showing lower. So it kind of illustrates, you've got to be very careful to know, well, what is my overall picture of health from all of the metrics, right? So you can put the particle count into some kind of context. And this was a prospective study following people over many years. So there was plenty of data here to show the trends that might be interesting. Here's another quick example of why I became interested in this question back in 2013, the Quebec cardiovascular study. And again, we see a similar kind of phenomenon, that high particle counts were a massive multiplier of risk relative to lower in this study. But interestingly, they stratified and saw the lower insulin people, there wasn't really much of a multiplier. And below 12 is not even that low in insulin. You should be around 5. So again, it points to something we need to understand more deeply to make better decisions in our lives. I did quite a bit of study for this coming material over the past couple of months. There's my desk as I'm preparing. and I had to use other desks because I had nearly 200 papers on endothelium and glycocalyx in the end. But they all came together and gave some interesting data, which I'll share. So here is the atherosclerotic process. We've got the LDLs coming in through your endothelium into your artery wall. This is the artery wall. And then you get an inflammatory cascade that ends up with macrophage engulfing and foam cells forming and artery wall swelling and eventually ruptures and breakouts and heart attacks. But we're not going to really go through the inflammatory cascade because it's an established science and it won't add a huge much value here. But I will note that the target list of key things that the Semaine and Kitavans achieve, for whatever reasons, will help enormously with moderating this inflammatory cascade. So it's relevant here, but we're not going to go through all the detail. We'd rather focus on the core of the LDL hypothesis. And that's shown here, that if you have more LDLs, more will get into your artery wall and kick off issues. It's not quite as simple as that though, because there are layers of evolutionary design technology, essentially in your body, which are there to make sure that your LDL particles, whether higher or lower in number, generally speaking, do not pose a threat to your health. And that would make sense. Evolution is not an idiot, after all, as I like to say. 1, 2, 3 layers, and we'll go through them each briefly. So one, you've got the glycocalyx, and this is an extraordinary structure of proteoglycan type molecules, these little blue hairs here, and they grow essentially on the inner wall of your artery. And in here is the central flow in your artery with a red blood cell. But we've got your LDLs in your blood flow, and the glycocalyx, it's becoming apparent, has a sieving function on managing LDLs in the bloodstream. And also platelets are prevented from reaching the arterial wall, and inflammatory molecules are prevented from reaching the arterial wall and causing issues. So the glycocalyx is very important. They didn't know about it until 30 years ago. Then you've got your endotheliums, your endothelial cells. So this is a single cell layer. They're bound together with tight junctions on the very inner wall of your artery, and that's a sieving and management of what goes in and out of your artery wall. So that's layer 2. And down in your artery wall, in the intima, we've got proteoglycans, and these are not too dissimilar from the glycocalyx structures. They're amino acid and glucose kind of strands, and they do catch LDL particles down here, entrap them, and then they can get managed, ideally engulfed, and ideally the cholesterol transported back out by a high density lipoprotein and other mechanisms, if you don't overburden the system, that is. So 1, 2, 3. Now we would be interested, of course, if you've got a higher number of particles, you would want to know what determines the rate of this 3-layer leakage. What governs, what mediates, what will drive more LDL down into the artery wall, and what will actually help the system prevent a disproportionate amount of LDL becoming entrapped. I mean, that's the million-dollar question. We can't just look at the number of LDLs being 1400 versus 900. 1400's bad, 900's good, but it's going to be completely dependent on this 3-layer system. So glycocalyx, layer 1. Here it is, an electron microscope graph or photo, and it's pretty small, a couple of 100 nanometers. And here's a paper I'll just mention out of the many I got. Arterial glycocalyx dysfunction is the first step in the atherothrombotic process. That's kind of a strong title, and it was a very good paper, but it would be controversial because the first step is generally seen to be just LDLs going through the wall and getting stuck. So an interesting paper if you want to look it up. Here an outfit, and I'm not connected to them, did high carb meals in people to look at what happened to the glycocalyx. They didn't give full details, but I'm reckoning they were high sugar too. So there's the normal glycocalyx, 2 hours after the meal. 4 hours and 6 hours. And as you can see, there's a dramatic impact, no doubt about that. They noted that it takes the body 8 to 12 hours to fully recover up to the healthy glycocalyx. And of course, most people will be eating too regularly. They'll be eating high carb meals and they'll be dropping another stone down into this pool within 3 or 4 hours. So you can probably guess the glycocalyx has a hard time of it. On the left, we see the glycocalyx. Here's the hair-like structures, and there's also a recently discovered boundary layer that also exists, and that keeps the red blood cells from the artery wall. It manages to an extent the LDL interaction with the arterial wall, and it manages inflammatory components that could inappropriately be interacting with the wall. On the right, we have a glycocalyx in a mess, a bit like we showed a minute ago. This person might be smoking cigarettes or drinking Coca-Cola. We don't know, but you can see that things are not so good. Quite an old paper looked at this, and they did laboratory tests, not in humans, so we need to take them as they are, laboratory grown endothelial cells, and they tried to attack the layer on the cell. And normally they didn't get much LDL, lipoprotein internalization, into the endothelial cells in their petri dishes. But when they damaged this outer layer, right, with agents, they got a much higher rate of LDL getting to the endothelium and incorporating into the cells. So that was interesting. When they went ahead with cationize ferritin, a very aggressive agent, they noted that they reduced this layer generally to maybe 5% of its original kind of thickness or robustness, and they likewise saw a much higher increase again in the LDL getting to the endothelial cells. So they were demonstrating a form of barrier function to this glycocalyx. The other papers I have, to move briefly, high sugar, high fat diets would impact the glycocalyx, hypertension and hypertension related signaling molecules, oxidative stress, oxidized LDL in the blood, not native LDL it would appear, smoking certainly blows your glycocalyx away, and arterial morphology. So we know that we get atherosclerosis in a focal sense. It's focalized or localized to branch points of the arteries in most humans. And now they're discovering that the glycocalyx there is very much reduced in thickness and beginning to tie that to the focal nature of atherosclerosis. And finally, ischemia reperfusion after operations, where you basically intervene into a human. They've noticed rapid atherosclerosis in replaced arteries in the short period following such operations. And I have some papers now linking that to the destruction of the glycocalyx, which is inevitable relating to the operation. So loads of fantastic science out there to be explored in coming years. But again, all the things I just listed, which are negative for glycocalyx, they'll certainly relate closely to our list of key things you want to keep an eye on to keep your arterial health. So there's quite a lot of overlap there. Layer 2, the endothelium. So this is one of nature's wonders. And it's basically a single cell layer on the inside wall of your artery. Up in the middle of the artery flow lumen is where the blood and the LDLs are. And on the other side of the endothelium, you've essentially got into your artery wall or your intima. Now I'll first mention endocytosis. Don't be worried about the word. It just means that the cell, for its own needs, calls an LDL particle using an LDL receptor, engulfs it into the cell, and breaks it up and takes out its components. And this happens in myriad cells all over the body. They bring in the lipoproteins and take what they want from them. And Brown and Goldstein won the Nobel Prize for discovering the LDL receptor and some of these mechanisms. But endocytosis is not what we're looking at today, because the LDL does not go through to the artery wall. It gets used internally. But I just wanted to let you know what that was. And this study, really briefly, they just put up the concentration of LDLs in the blood flow by a factor of 10. And they noted that the endocytosis rate did not really change because the cell is deciding how much it wants to endocytose. So just an aside. More interesting for us is transcytosis. Now, as you can see, this does bring LDL particles complete into your artery wall from the blood flow side. Now we're on the money. And it does it through different receptors than the LDL receptor. There's question marks here because my most recent papers in 2018 are actually marveling at the fact that we don't fully understand this process yet. We know it happens, so they're working it out, and we'll keep an eye on them. Here's a paper, though, quite some time back, which labeled LDL particles with gold, and the gold was to be able to track the LDL through the endothelium. And after a few minutes, you can see here, they actually had LDL lipoproteins with the gold attached in vacuoles in the endothelial cell. So that was fascinating for them. And after some more time, I think just a matter of hours, they had LDLs with the little gold particles right across in the equivalent of the arterial wall. So they had crossed the endothelium fully, the endothelial cells, and brought their gold with them. They were kind of like little Irish leprechauns carrying their little pots of gold across the endothelium, right? So sorry about the stage Irish there, but so yeah, but the point is important. They come across the cell complete. And yeah, they saw around 25% after a certain period were fully across. So that kind of tells you this process is very efficient and evolutionary and managed. It's a designed process. It's not a mistake. This doesn't happen by mistake. Transcytosis from a paper to give an idea, if you have 10 times the number of particles up here in the blood, you get around twice the amount of transcytosis. So it's not proportionate, it's a moderate amount of extra LDL coming across if you really put up massively the number of particles. But interestingly, there are other papers showing what drives transcytosis way beyond the particle numbers. Here they had TNF-alpha, an inflammatory component, which is very active in inflammation, insulin resistance, diabetes, and many other inflammatory conditions. And when they brought in TNF-alpha, they brought up the transcytosis of LDL particles by a factor of 4.5. So that was actually more dramatic than putting up the LDL particle number massively. You know, TNF-alpha was very potent. And your brain, just as an aside, I've heard it said the brain doesn't take in LDL. Well, it is true that the brain produces its own cholesterol locally. So I think the astrocytes. So it doesn't really need LDL, you might say. But this paper, interestingly, showed that the brain does take in LDL in small amounts with the LDL receptor. It transcytoses, which is kind of interesting. And when they depleted the brain cells, this team, of cholesterol, they stripped them of cholesterol, brought it low. They were wondering, would the brain take in more LDLs based on this? And they did. They upregulated the LDL receptor, these astrocytes, and drew in quite a bit more LDL. Now, these are still small fluxes. You know, there's not a whole lot of ApoB, lipo or protein in the cerebrospinal fluid, but it's still interesting evolutionary designed the system to work. Other papers which drive higher transcytosis, which you probably wouldn't want if you've got quite a few particles up in your blood, right? C-reactive protein, oxidized LDL, oxidant induction generally, radical oxygen species, TNF-alpha we mentioned, and lipopolysaccharide. So bacterial components, often from leaky gut or maybe from periodontal disease, those components in your blood have been demonstrated to drive up this transcytosis as well. So all these are bad things. But again, they're quite linked in many cases to our list of key things to keep right in your body. And again, this list is not exhausted. See the etceteras down here. There are many other things, but this is just a pretty good rule of thumb list, top Pareto items to keep your eye on. There's paracellular transport also across the endothelium. And this is leakage, for want of a better word, of LDL particles across from your blood through the barrier and into your artery wall. And there's a lot of tight junctions in there to prevent this leakage or manage it, but they can become somewhat affected and dysfunctional. So you want to avoid that happening. An example again involving TNF-alpha here, they put in TNF-alpha and they raised by 4.5 times the amount of LDLs coming across by this paracellular pathway. And the guys worked out as well why, because basically the apoptosis rate of the endothelial cells, or the death and turnover rate, was linked directly to the LDL permeability number. So essentially the TNF-alpha was causing endothelial cell death and turnover, and during this turnover process there was a lot more LDL allowed in. And again, this may be an evolutionary design that when you've got damage to the endothelium, there maybe is some requirement to do this. But that will be found out in coming years. So other papers, not too many on this. What causes increased paracellular transport? TNF-alpha, angiotensin II, which relates closely to high blood pressure, and generally any insult to endothelial cells will cause turnover and will increase this vector. And needless to say, our target list of things to watch, right, that the Kitavans get right, that would generally speaking link very closely to things that would drive up paracellular transport of LDL. Layer 3, nearly finished, the INTIMA PROTEOGLYCANS. And these are the proteoglycan structures in the wall of your artery, and if LDLs get in, they kind of entrap them due to charge, and they hold them there to be basically engulfed, and hopefully the LDL will be taken apart and the cholesterol will be taken back out by HDL if your HDL is very functional and the efflux is working well. But in people who have problems with that and are driving a lot of LDL issues, you know, the system will be overwhelmed and we get the atherosclerosis accelerating. But anyway, these are the proteoglycans. But we'd be interested, what kinds of things increase the LDL reactivity and binding with these proteoglycans? Given the same number of particles, you know, what would drive them to bind more and get caught up more and maybe not just go back out again? Well, the papers that demonstrate this, blood from heart attack patients does this, much higher binding, blood from insulin resistant people, blood from type 2 diabetics. And this is with cholesterol at the same levels, it's just uniquely their blood and the lipoproteins or LDLs in their blood have something different about them that causes them to get caught up here. Small, dense and oxidized LDL, certainly higher affinity to bind, and especially with higher ApoC3 content, which we associate mainly with insulin resistance and diabetic type problems. And small dense LDL also show a higher affinity, and that also relates. And an interesting mouse experiment, they got type 1 diabetic mice versus wild type controls, and they wanted to see, well, why does type 1 diabetes cause so much more atherosclerosis? And they were specifically looking at the binding down here that we're discussing. And it turned out the type 1 diabetic mice with the same cholesterol levels as the wild type, they stabilized that, had 8 times more retention down here due to the nature of their LDLs and the nature of their blood. So that was quite fascinating too. Generally speaking, not exclusively, our key list target things will also tend towards, from what I've seen, towards more reactivity and binding down here in layer 3. So our list kind of mediates to a point all 3 layers of this managing of your LDL. So 1, 2, 3 layers, and all of them, to some extent, yet to be fully determined, are affected by our target list and more. There are obscure inflammatory conditions which could also cause damage here and a change in flux. Okay, so it's important to be thinking about these when you're thinking about the particle number. There's one bonus layer, and I'll finish on this. I think I'm going pretty long on time, and it's oxidized LDL as a cause, right? Now, this is a bit of a heresy. Mostly it would be suggested that the mildly oxidized LDL in the blood that happens due to your diet and lifestyle doesn't really matter so much. But let's look at it, because there are published papers and I think we need to discuss the science, always. So this paper was quite interesting, but it was a little heretical because as you can see here, it suggests that the oxidized LDL, minimally oxidized in the blood, right, is the thing that causes endothelial dysfunction, one of the big things, and does come in, right, relating to that dysfunction, and gets engulfed with the LOX receptor and becomes foam cells, blah, blah, blah. Okay, it sounds reasonable. But the heresy is that this study team indicated they didn't believe LDL native, undamaged, non-minimally oxidized really had much to do with the process of atherosclerosis, and that is heretical. So we look at a couple of papers. I went digging to try and see is there any data around this, and this team got healthy controls and people with very high LDL and bad ratios, and these people were patients too. So we don't have all the data for them, but they expected them to have issues. They took their LDL samples, and in each group, healthy and unhealthy, they had L1, L2 and L3 fractions of their LDLs, and these are non-charged, pretty much healthy, non-oxidized LDL. They noticed though that they picked up quite a bit of L4 and L5 in the unhealthy people, and L4 and L5 are minimally oxidized, so slightly oxidized on the outer shell of the lipoprotein and the apolipoprotein itself. And they looked and said, how will this affect endothelial cell health, or cell death is what they were looking at, percentage cell death in a preparation. And the L1, L2 and L3 didn't have any real effect on the cell death, right? So native LDL didn't really do much to the endothelium at all. But L4 and L5 from the not so healthy guys, that caused very high levels of endothelial cell death in culture, which is interesting. And they went ahead, this team, and they used copper to oxidize the hell out of LDL particles just as a control. And interestingly, yeah, it caused endothelial cell death, but not much more than the L5 from the kind of semi-diabetic guys, I would say. So that was an interesting observation. And we know that oxidized LDL in the blood is, it's called minimally oxidized, just what we see here, is a very strong risk factor for future events. So tying this together, you'd say, maybe the heresy isn't so heretical. Maybe there's something in this. One more heresy, just a little to finish you off. So this study was a Russian team, 2017, the last one was 2009, so this one's bang up to date. And they're looking at desialization of the LDL, which is a step they describe as just before minimally oxidized. And they say that this desialization in itself is the early step that causes LDL to be a problem. Are they correct? Well, they did a good study. They got healthy controls, type 1 diabetics and type 2 diabetics. They took out their LDL fractions and they saw that the healthy controls at a very small level of desialated LDLs, type 1 diabetics had more and type 2 diabetics had more. So probably not too surprising. They looked at the accumulation of LDL in the intima, and they actually took intimal cells, smooth muscle cells and other artery wall cells from cadavers, from people who died from car accidents and the like. So they made up a preparation of arterial wall, a human one, which is pretty good guys. And they saw that the non-desialated, LDL did not really have any magnifying effect on the accumulation of LDLs in the arterial wall preparation. But when they separated the desialated LDL from these people, we could see a big multiplier, right, of accumulation of LDL in the artery wall cell preparation, right? Quite dramatic. And increasingly as you go up from normal to type 1 to type 2 diabetic blood. So they did come to the conclusion that this was very important, and perhaps was important in terms of the health of the LDL particle even before it becomes minimally oxidized and can be picked up in the ox-LDL assay. So again, the heresy has to be entertained. And again, these are only some papers, pretty recent, so I think in the future all of this will get debated and argued openly, as all science should be, to come to a firm conclusion on it. So we're finishing up now. Layers 1, 2, 3, evolutionary designed, exotic and amazing, that can be very much affected by some of the primary root causes that myself and Dr. Gerber go through in our book. And they're not the only root causes, there are many more, but you got to do a Pareto principle and stick to the top things to work on. You can't work on everything. So we believe in the world. This list covers a huge chunk of what's causing all the chronic disease in the world. So it's not exclusive, but it's a big chunk, right? Focus on the big stuff, big bang for the buck. We also brought up oxidized LDL, which, if the theory bears out in coming years, could be a very significant problem in this whole atherosclerotic milieu in itself, independent almost of these three layers, but certainly probably contributing to problems with the three layers. So synergistic effect, perhaps. And just to keep in mind, you should keep on target. When we're looking at something like the number of LDLs, it's one single factor, and it may be very dependent, a higher number, on whether you've got myriad other issues or myriad other possible issues that allow the higher number to translate into a significantly increased risk. So this is just something we should keep in mind. And especially in engineering, we always look at all the variables, how they interact together, right? How there can be synergies and how there can be dependence and independence of variables. Some things can cause disease acceleration in certain circumstances, but those certain single things may not cause significant disease acceleration in other circumstances. And it behooves us to always look at the whole system complexity and the full picture in order to come to any conclusions. I will say, however, and this is just our book, Eat Rich, Live Long, published 6 months ago, I will say that if your LDL particle count really jumps up on a new diet, then you've got to be careful because it is a known risk factor. It may not be independent, but it still is a risk factor. And there are easy ways for you to bring the LDLP number back down, and we go through them in the book. You know, you eat a little more healthy carb, as opposed to being ultra low carb. You swap sat, fat and protein, maybe from animal products. You switch over some of that to monounsaturated, healthy real foods. And we have a list of around 10 things, but you can actually bring the number back down. Now, whether the number for you, if all your other metrics, ratios and everything else is good, whether the number really means a lot for you is hard to be certain on. So if you wish, you can bring it back down relatively easily, and that's fine. It's an individual decision for everyone. We believe in free will and the American way. So I hope you enjoyed that, and don't forget to sign up for Adapt Live and see more crazy Irish guys and guys from all over the world actually having hopefully very interesting chats with you and filling a little piece of your day with something useful. So thanks a lot, Adapt Live, and so long from Ivor, Fat Emperor, and I'll catch you next time.